for those introductions. And as Emma said, like, I think taking an interdisciplinary approach to everything, but specifically kelp, has been so important to me in my journey and what I've experienced in regards to restoration. And so this is going to be totally a jam-packed like talk of lots of different things. Hopefully I take you through it in a way that makes sense, but the premise is like an interdisciplinary approach. Um, so yeah, I'm just really excited, to be here. I've had the privilege of giving this talk once before at Scripps, and that was really fun and got some pretty cool feedback. And that's how I got to give this talk today. So I hope you like it. But yeah, let me just organize my screen real quick. Okay, here we go. All right, so I'm Abby, and I graduated from Sonoma State University with my master's in December. And now I'm Assistant Diving and Boating Safety Officer for UC Davis, based in Bodega Bay. So like Northern California, maybe the lower limit of what some people call Northern California. But throughout my time studying kelp, I've just met a lot of really wonderful people and learned a ton. And I've seen a really incredible community form around kelp. And so I just want to like start off by acknowledging how important this community is. And I think that this kelp culture talk and just the series really illustrates like the community that we do have. And, I've seen people on here who are artists and who are scientists and who are just like community member enthusiasts, people that run nonprofits, people who are researchers, everyone from different backgrounds. Has developed some interest in kelp, I think. And it's really cool to have this community of people that, we're not just excluding people if they're not scientists, right? It's like so many different people bringing lots of different perspectives. So I've had the privilege to work with a lot of you and a lot of people from different places, but I think, everyone has a piece of this like kelp puzzle that they're specifically interested in. And that goes a long way for restoration, right? Like we can't all just tackle everything on our own. So like bringing in this interdisciplinary approach to all of these projects has not only been fun, but I think what's made it really successful. So I'm going to start off with an introduction of myself and my background, and then we'll kind of go through some research techniques that I've found to be super helpful for the research that I've been involved in. So I graduated from Whitman College in 2020 with my Bachelor of Arts in biology. And so right off the bat, I've been interested in like interdisciplinary education, specifically focused on outdoor education. And my college experience really inspired my love for just teaching people about the outdoor environment and leadership and empowerment combined with science. So that's kind of where I get my foundation from. And then I went on to become a scuba instructor where I got to teach kids and teens in the Pacific Northwest. I'm from Seattle, Washington. And so I got to teach them up there. This is a photo of, they're a bunch of 10-year-olds actually that we took scuba diving. And it was just so cool to see the impact that like teaching scuba and leadership for these kids had on just their confidence and awareness as individuals. So that just like, I love, teaching these sorts of outdoor activities. And then I also am into underwater photography and videography. And so that's a big component of my job too. But throughout all of these different areas, really the main focus is just educating people about the ocean and engaging them. And I just love sharing my experience with others because I think it's really awesome. So I've also have the privilege to dive in many different places. And I'm really passionate about the work I do. So I got to dive in Antarctica as a scientific research diver. I dove for Sonoma State University as a research diver doing kelp work. And now I'm at UC Davis, like I said, as assistant diving and boating safety. So I get to work with a lot of different people, a lot of different agencies, different research projects to really merge all these things that I love into work that is supporting both our ecological and our social human communities. And then also throughout this, like I've merged my role as both a student and a teacher, which I think is really important. Seems like we have a lot of people on this talk today who have varying backgrounds. We've got some professors, we've got some students, we've got people all over the place. And so I just really encourage you to carry that student mindset with you throughout your journey, wherever you're at. And I just really try and embrace that to just continue learning. So With that being said, I hope that you all could just take a deep breath here. Thanks so much for giving me an hour of your time. And for the next 40 minutes, we're gonna just hopefully learn something together. And I'd really like you to just maybe think about science a little bit differently than you might normally do every day, especially if you're an academic. Um, and I hope that there's something here that you can walk away with that maybe you're feeling extra excited about or inspired or empowered. So this is where I want your mind to be at, hopefully. This is where my mind is usually at. Anyways, okay, so research techniques. So I've identified 3 research techniques that have been, super important to me in my journey as both a scientist, storyteller, and a leader as well. And so we're gonna break these things apart, and I'm gonna teach you about some storytelling techniques and how that's been important to kelp restoration, the actual science of kelp restoration that I've been a part of, and then the teamwork that goes along with it. And I think these 3 things are really important. And I know that it might seem kind of basic, like, okay, like why are we just really breaking this into simple components? But I think it's important to think about things like simplistically, and you'll notice that science is only one part of what makes research really important. So without further ado, we're going to talk about storytelling first. So this is a photo that I took when I first got my underwater camera. And it was an honorable mention for the Hilda Cantorlund Phycological Society Photography Contest, something like that. Really long title. But basically, I titled this photo as Hope for the Kelp Forests because to me, this photo represents the future of bull kelp. And so this is a bull kelp saurus sinking to the bottom. This was taken in Carmel in California. And this is the saurus that settles on the bottom and is providing the next generation of kelp to the forest. So I'm kind of assuming that a lot of you on here are pretty invested in kelp and kelp science. And so hopefully you don't mind me using a little bit of jargon. But yeah, feel free to drop any comments in the comment section. And I might try and check them throughout, or maybe Jess and Emma could answer any science questions or other things that might come up. So for storytelling, I want to ask you, what is the main driver of research? You can feel free to drop it in the chat. Just think about it for a second. What drives maybe your research or other research that you've seen, whether or not you are a researcher yourself? I think this is something that we all think about. So go ahead and take a second to think about what's the main driver of research. Since we're not a live audience, I will just have to tell you the answer. Let's look at the chat here. Curiosity. Oh, you guys are this is so sweet. Yeah, curiosity. Yep. Problem solving. Okay. See, those things are all awesome, right? That's what we think the driver of research is. But really, I'm going to say money. Money is the driver of research, realistically. I think that the questions are important, but I think for a lot of you scientists on here, the research is not going to happen without that funding. And so in order to get the money and support to fuel that curiosity that you have to ask all those questions, you need to convince funders and the public that your cause is important, right? And the best way to engage people is through storytelling. And luckily, good science is also going to tell a good story. So let's check this out. So this is, you might have seen this. This is like the hero's exposition or hero's story arc or whatever, plot development. So in just like a typical story, we have the exposition where the background gets laid out, we set the scene, and then there's some sort of rising action or conflict that happens. And then there's like the climax of that, the falling action, and then the resolution. And so it's just this nice, really beautiful arc. And that's like a classic throughout most stories that we experience today in the media, books, movies, etc. But if we like superimpose that onto our science, we kind of get the same arc, right? And so when we're doing scientific writing and we're writing a scientific paper, we start off with a background and we kind of meet people at where they're at, what we think is important, set the scene, and then we identify some problem that needs to be solved. That's a conflict because we don't have the answers yet. And then like that climax is going to be the methods, like what did you do in order to resolve that conflict? And then you've got the results of the study and then the conclusions. And so, yeah, if you can think about this in your scientific writing, that at least has really helped me engage people with writing grants and proposals and giving talks and things like that. So in order to tell compelling stories, we don't just use writing though, also we use photos. And so just here's another one of my photos that I've taken that is one of my favorites because I think this photo really speaks for itself and it tells a story in itself. And so I found the photos that I'm really excited about and things that people have engaged really well with have been the ones that actually tell a story. So this here is bull kelp that is being eaten by purple urchins. And so I often use that, to set the scene for this problem that we're having with our bull kelp in Northern California. So photos and videos are going to help engage people with your story. And so hopefully I don't have to convince you too much the value of photos. But I think the value of being a scientist and an observer and an artist and photographer, all of those different things, however you identify, is really important. So coming from a science background and combining an artist's lens, I think it's really cool because that can make science more accessible and interesting to people. It allows you to communicate your work better. Photography can also be a way of collecting data as well and making those observations. So I'll show you a few examples of how I've personally used photography and science, combining those lenses to make cool observations. So this photo is of Desmorestia or acid weed and in a bull kelp forest. And this is in Mendocino, California. And this photo is actually the premise of my master's thesis. And so Jess, if you're on there, if you could drop that link I shared with you, please, in the chat, that would be awesome. But I conducted my master's thesis on the role of Desmorestia in bull kelp forest, specifically with the idea that it may provide some sort of protective barrier against urchin grazing and serve a really important role in the margins of the kelp forest. So you can check out that work if you're interested, but that was totally inspired by the observations I made through photography. When I was looking back at my photos, I realized this algae was popping up everywhere. What is it? What's its function? And then also, this is one of my favorite photos. It's not a great one, but I just always have a little camera on me while I'm out diving and doing work. And you can see this is bull kelp growing on a Pterogophora stipe. And so it's like elevated up off the substrate in, this kind of barren area. This is at Van Dam in Northern California. And this is really cool because this is kind of a spoiler alert for what we've been doing for our most recent and successful kelp restoration projects. So thinking about how that bull kelp is elevated up off the substrate. So yeah, this photo was like used to sort of inform those ideas there. So yeah, that's on combining lenses between photography, storytelling, and science. And so I'm gonna kind of tell you the story about bull kelp restoration in the way that I tell it. And again, I'm assuming that there's some background here with you all in knowing sort of what's going on with our bull kelp. But I'll tell it to you in the way that I often do to engage with audiences, and hopefully maybe you'll get some nuggets that you like or that work for you that you can take into your grants and project proposals or defenses or whatever. So here we go. This is how I usually introduce bull kelp. I say, this is how lots of people interact with bull kelp, right? Sometimes you smell it before you see it. You see it on the beach. There's lots of flies buzzing around it. It's an important ecosystem in itself, but this is not necessarily the way in which I interact with it. And the way in which I have grown to love bull kelp is by seeing it underwater like this. It's a really spectacular individual, but it also forms a forest just like our terrestrial forests, but underwater. And so it kind of creates the same function. It's got a 3D structure. It provides habitat. It provides, environmental factors that lead to really beneficial, habitat for other animals. And it creates this whole ecosystem. And bull kelp is, an annual species. It's unlike giant kelp, which is a perennial. And so that creates a different ecosystem dynamic. But yeah, bull kelp. And this is the bull kelp range. And so it has a pretty wide range in the Pacific. So we've got from Alaska down to Central California is generally where we find bull kelp. And a lot of my bull kelp research has been in Northern California, specifically Mendocino and Sonoma counties. And so this land is really beautiful, really spectacular. It, belongs to the Coast Miwok and Pomo Indians. Now I think generally referred to as the Kashaya Band of Pomo Indians, are residents on this land. And yeah, it's just a really beautiful and important area in the story of bull kelp. So I'm going to show you a video of the site that you see in this photo just underwater. And this video is, they were taken 1 year apart. So you'll see one video, and then the next one is going to cut into what it looks like a year later. And this is what we're experiencing with our kelp forest underwater. So here we go. I don't think you can hear my sound on here, but there's really dramatic music that goes along with this video because I think it's pretty dramatic and moving. So this is Big River Mendocino, or also known as Portuguese Beach, and that was 2022, and this is a video from 2023. And what we can see here is the widespread collapse of our bull kelp forests underwater. And the difference here is that there's no structure and very little diversity in life that we see in that after video. And our bull kelp forests have now turned into these urchin barrens because of these hungry purple urchins. And so these purple urchins are a natural part of the ecosystem, the bull kelp ecosystem. However, their relationship in the bull kelp forest has changed. And so in Northern California specifically, we've had two top predators in the ecosystem historically, sea otters and Pycnopodia, or sunflower sea stars. And we unfortunately lost our sea otters due to the fur trade in, I think, the early to mid 1800s. And so they've not been a part of the ecosystem for a really long time, which means that there was no trophic redundancy in the ecosystem. And so we really only had one top predator, one controller up there. And that was the Pycnopodia. And I just want to shout out the Pycnopodia really quick because what I like to call them is a gateway species. So maybe you have a gateway species that got you interested into kelp or whatever it is that you are interested in. But Pycnopodia are my favorite. This is my Pycnopodia tattoo. I love them dearly. They're super cool. They're the biggest sea star out there, and they're super fast. They have 24 legs. They are an incredible predator of echinoderms. And yeah, they're just really spectacular to watch. But unfortunately, we've lost them from Northern California. And so we've lost two of our main predators. That happened in 2013 and 2014 due to sea star wasting syndrome, which was an outcome from a warm water anomaly that hit the coast. We call it the blob. And so the loss of the predators resulted in a 95% loss of our bull kelp in Northern California. And so you can see on this figure, this is from a Laura Rogers Bennett and Caton paper from 2019. This is the Mendocino County and Sonoma County stacked on top of each other. And then on the left, you see 2008 kelp coverage, kelp canopy coverage in green. And then 2016 was the kelp canopy coverage. And yeah, that was the result of the warm water blob, subsequent loss of predators, and runaway herbivory of purple urchins. So we've lost 95% of our kelp in Northern California. Hopefully you are familiar with that idea, but one question that has been driving a lot of the research that I've been involved with is what about the remaining 5% of kelp? And my lab, particularly at Sonoma State University, Dr. Brent Hughes, we call it the Coastal Conservation and Ecology Lab. Oh gosh, I hope he's not on there because I think I messed up that title. But anyways, so we're interested in persistence of kelp and what is driving the change in kelp and what is potentially limiting the recovery of kelp as well. And we need all of these components in order to restore. So there are two ecosystem properties that I identified for my research that determine the response to change. And so the first is ecosystem resistance. And that can be defined as the capacity for the ecosystem to retain structure and function despite disturbance. So if you think about a rubber band, this is basically like how easy it is to stretch the rubber band. So if the rubber band's our ecosystem, the resistance is going to be how easy or hard it is to stretch that. Likewise, we have resilience, which is the ability for an ecosystem to regain function after experiencing some sort of disturbance. And so this is like stretching the rubber band and then seeing how it snaps back and regains its function after it's been stretched and disturbed. And so these two ecosystem properties are really important in defining how that ecosystem will respond to disturbance and change. But what happens when our rubber band breaks? And that's exactly what we've seen in Northern California. So there is a natural fluctuation of kelp in the ecosystem via the resilience. It will leave and be disturbed, and then it will come back until we ultimately break that rubber band. And so this is a figure from the California Department of Fish and Wildlife report that came out in 2023. And you can see it's a time series along the x-axis and then annual max canopy area of bull kelp on the y-axis. And that red dotted line is that marine heat wave event, that 2013-2014 event, which ultimately is what led to the collapse of our kelp. And that fluctuation has not bounced back anymore. And so we've kind of got this flat line of kelp in Northern California, which is super unfortunate, but it also has provided an opportunity for human intervention in this ecosystem because it's not having that normal resiliency, that ability to bounce back that it has in the past. And so that leads me to the research that I've been focusing on, which is ecosystem management through restoration, specifically bull kelp forests in Northern California. And there's two main ideas that we've focused on broadly for bull kelp restoration, which involves kelp enhancement, so bringing in kelp into the ecosystem again to restore it, while also suppressing grazers. And so those things that are eating the kelp and preventing the kelp from regrowing naturally. And so there's lots of big players in these projects. I've been really fortunate to work with so many different people all throughout the region, as far as like Humboldt collaborators all the way down to Santa Cruz and even further, Scripps, and so many different people and grants and projects that have been coming together to do this experimental restoration, which is kind of the phase that we're in right now. So with that, that's my bull kelp story that I like to tell people to introduce them to the restoration and the projects that we're doing. So the next part that I want to get into is the research technique number 2, which is the actual science that we're doing to restore bull kelp. So I wish that, we just talked about storytelling. I wish that my photos alone could bring back the kelp. But yeah, we do need the actual science to do it. So, photos and storytelling, super important to engage people in what's happening. But then there are some people, like probably many of you, who want to know about the nitty-gritty science. And so we're going to get into that. So Northern California kelp restoration can be pretty challenging, as you can see from this photo here. Pretty low visibility. Let's see. So we've got some challenging coastal access. It's really hard to access a lot of the beaches in Northern California to get down there to do the work. High wave action, big swells, low visibility, as you see in this photo, is pretty regular for up here in Mendocino. Pretty cold water. We've got that upwelling, ranges from like 48 to 56 degrees, I would say, year round. Relentless grazing pressure from our purple urchins that have established themselves in these barren states, and they just won't stop eating anything that's out there. There's a high effort required for these urchin removals. If we want to take them out of the ecosystem, they just keep on coming. So you need a lot of money, time, people to do that. There's also big biters up here in Northern California. So we definitely have to think about sharks in our research and what areas are safe to access and what otherwise might not be. I'll get into that a little bit more later. Diver availability, who is actually able to do the work underwater, can sometimes be a challenging thing to navigate. It's not as popular being up here as it is maybe the warm Southern California. So Also, we've got timing and seasonality of our annual bull kelp forest. So as I mentioned before, bull kelp is different than giant kelp because it doesn't exist all year round. It dies in the winter, and that's part of its normal life cycle. But it also makes it hard for restoration because we need to think about this timing and really time our restoration with the natural cycles of bull kelp. And then also kelp has two different life forms. And so when we're thinking about restoration and management, we have to think about how do we manage both of these different phases that the kelp goes through? This is a pretty common consideration for fish and for invertebrates in how they'll have like a pelagic life stage or a nursery life stage, and then they'll go into the open ocean, things like that. But kelp has that too. And so again, I hope that maybe you're familiar with the bull kelp life cycle, but if not, here's a little diagram that my friends Rachel Carmen and Julieta Gomez made. And you can see we have a kelp sporophyte, an adult that has these reproductive sorrel patches on them that have the spores. It settles and then turns into gametophytes, will mate with each other on the bottom of the reef, and then they'll stay dormant throughout the winter and then grow in the spring and summer when the light availability is there and the water's warm enough. So this kelp, they have free swimming phototactic biflagellate spores, which is quite a mouthful, but basically means that the spores kind of have this like free stage where they're settling down into the substrate and then they anchor to become that sedentary individual. And so my research work specifically has focused on the experimental design of restoration methods for growing bull kelp in the lab and then transferring it out in the ocean under the idea of conducting kelp enhancement for restoration. So I'm going to talk to you about some of the techniques that my lab has used at Sonoma State University, but in collaboration with specifically Moss Landing Marine Labs, and lots of other individuals from different universities and institutions that have been working on this as well. So I heard someone in the beginning mention green gravel, which is a technique that has been tested for bull kelp and giant kelp restoration. Sorry, giant kelp restoration and other kelps, but we were testing it for bull kelp on the North Coast. And so that's just growing kelp, again, in the lab on certain substrates, utilizing their life cycle. We can kind of harness that to then grow it on whatever we want and then plant it out in the ocean. So for green gravel, we were growing the kelp on little rocks, and we outplanted them into the ocean and found that there was a little bit too much wave action on the North Coast to really get these rocks to stay where we wanted. Although this technique has been shown to be successful in other places. I think Norway, potentially, don't quote me on that, but I think Norway, it's been successful to use green gravel for planting alaria or other kind of long benthic kelps like that. But it did not work for us. And then we also learned the importance of timing for getting these kelps out. So like I said, the kelp with its annual life cycle, it's really important to think about when it would naturally be popping up or settling and what time it's appropriate to get those out plants out into the field. Do you get them before their dormant phase or do you put them out after the dormant phase? Something we're still thinking about. But then we tried these pavers. And so we tried growing the kelp on these little rocks that we grew in the lab and then bringing them out in the ocean and like epoxying them to the ocean floor. And so these pavers were not the most efficient use of space in the lab because we needed these like trough tanks to be able to spread out all the rocks and you can't stack them on there. So it was pretty tough to be efficient there. And then again, also relearn the importance of timing and when are we going to actually grow the kelp on the rocks, which takes a while, and then plant them out in the ocean. So that's kind of where we got with the pavers, still in testing mode. But we tried also these seeded bricks. And so this is my friend Vinny wrapping twine around a brick. So we grew kelp in the lab on twine and then wrapped it around bricks. And I mentioned earlier the big biters that we have out here on the North Coast and how we do have to think about that for kelp restoration and navigate those big biters. And so I got to be part of this project where we were trying to do remote kelp restoration that didn't involve diving. And so we tried to grow kelp on this twine, wrap it around bricks, and then throw it overboard with lots of permitting so that, the forest could reestablish itself. So we're trying lots of different techniques in varying levels of like commitment and maintenance. And so this was like relatively low maintenance, just kind of throw it out and see what happens. But we learned that, yeah, this could work potentially for non-diveable areas. You have to consider the substrate that you're putting these bricks on, knowing that you're not going to be hand planting them out there because you're doing it from a boat. And our experiments from this study were inconclusive, so definitely something that could still be tested. Don't have results on this, but just an idea that we were playing around with. Then we've tried spore bags. So instead of in the lab taking the sori and then releasing the spores onto a substrate, we decided to take the spores and just put them directly out in the ocean in these bags in hope that they would just settle there and then grow themselves without us having to, outplant them specifically. And so with these spore bags, this is kind of the quickest and dirtiest technique. It doesn't require a lot. You just have to harvest the bull kelp sori and then put them in bags on some sort of structure. So it's pretty simple as far as restoration for us goes and things we've tried. But there is a signal. So when you can see the kelp growing, that can be anywhere from 2 months to a year, right? So thinking again about the bull kelp life cycle and how it's an annual, it could, depending on when you outplant the kelp, it could pop up within 2 months where it just, settles right away and then grows. Or you might not see if it worked until another year later. So that can be tough for restoration and doing surveys, making conclusions. And then also because these spore bags are attached directly to the substrate, the urchins can eat them pretty quickly. And so there's a photo in that bottom left corner. It's kind of hard to see, but there's urchins just like all over the bag. They totally get in there. So it can be a little tough, but still testing this technique out to see if there is a signal, if we do get a signal from these spore bags. And then most recently, we've tried seeded twine elevated up off the substrate. And so this is a new technique to bull kelp restoration in the area that I work in. Again, really only speaking on behalf of like the Mendocino and Sonoma bull kelp projects that I've been a part of. There's lots of other restoration projects happening all throughout the world and the country and the California coast specifically. But this isn't necessarily a new technique because a lot of seaweed farmers use this as well. And so we learned a lot from our seaweed farming friends who taught us how to grow the kelp on the twine. And if you recall back to when I was talking about the storytelling and using photography to combine lenses between science and art and making new observations, if you remember that bull kelp growing on the pterygopher stipe when it was elevated off the substrate, this was the idea with this restoration technique, was building an underwater infrastructure that would help elevate the kelp off the substrate where the urchins are to kind of create this refugia for the kelp to grow when we outplant it. Idea being that they would then grow into their full form and then settle on the floor. So this technique for seeding twine requires underwater infrastructure to be built, which can be pretty hard work, putting in buoys and lines and things like that. Again, thinking a lot about permitting and all of the processes you have to go to propose this work and do it in the correct areas. But it does provide a greater refugia from those urchins, which is really nice. We also put copper around the edges to prevent herbivory. That's a technique that a lot of gardeners use to prevent like snails and things from getting into their plants. We were able to use that underwater too to prevent snails and urchins from climbing up on the lines. And there is a potential for high yield for this technique. If you recall back to the pavers that I was talking about, the little like small stepping stones that we put out there, not so much potential for a high yield because you can't grow so much in the lab. But the researchers that have been working on seeding twine have really honed in their technique for making it incredibly efficient in the lab, getting as much kelp to grow and then spreading it out over these lines that could potentially lead to a lot of individuals growing on it. So like I said, this is kind of the newest technique that we've been trying in the history of my lab specifically over the last like 3 years of doing this work. But it requires a lot of effort from the individuals taking care of the kelp in the lab all the way to putting it out and then maintaining those seeded lines and making sure that things don't eat them or grow on them, constantly out there checking on them and making sure that they're okay, like any good farmer or gardener would do. Also that high spatial efficiency, so you can get lots of kelp on these lines, which is really neat. Thinking about like what size area we want to focus on restoring, what our goals are. But also with this seeded twine and restoration of bulk kelp in general is thinking about having a multi-year plan for the restoration that's taking place. And so one question that I like to pose to people is, what does restoration mean to you? What does restoration look like? What's the end goal, right? How do we define restoration of the ecosystem? Is it when we grow one individual kelp, or is it when we have the entire forest there? And I don't particularly think there's a right answer, but I think that's important in formulating our story and developing our goals for our science in our wide scale collaboration. And so for this technique specifically, we have the kelp growing on the line, which is really great, but it's not attached to the substrate. So the way that I've personally been thinking about it, and I think the grants have also defined it, is that restoration will happen when that kelp is reproductive, it settles, and then it actually attaches to the substrate. And that won't happen for at least a year after outplanting the kelp. So thinking about the duration of these projects has also been a challenge, but really cool piece of the puzzle. So as I mentioned, that's the newest technique and most current technique that the people I'm working with have been using. So I just want to highlight specifically some of those research projects that are happening right now. And so this is from last year. And this is Rachel Carm. She is the research technician for the Hughes Lab at Sonoma State University, but works in partnership with the Greater Farallones Association, who work in Sonoma County, as well as Moss Landing Marine Labs based in Monterey, but working all over the Sonoma and Mendocino coast. We have other collaborators at Sonoma State University, and then a lot of this laboratory work and culturing takes place at Bodega Marine Lab or Moss Landing Marine Lab. So I'm at Bodega Marine Lab right now. It's pretty cool to have this like little kelp farm in our backyard, which is pretty neat. So yeah, Julieta Gomez and Rachel Carm. Julieta's from the Greater Farallones Association, Rachel Carm, Sonoma State. They work together to grow these kelp in the lab. Oh, actually, let me go back. This is Rachel working in the trough tanks. And what's really spectacular about their work here at Bodega Marine Laboratory and inspired by the Moss Landing crew is that we have this annual supply of bull kelp culture that we can depend on for restoration that ensures that we can time the restoration correctly. So what that means is that we can culture bull kelp all year round when normally the bull kelp would die off in the winter. And then we wouldn't have access to kelp to start growing it at the correct time we need to be able to outplant it. So by having a culture at the lab that is cycling year round and always growing and always producing, it allows the researchers to be able to better time when they're going to start seeding their substrate and then when they're going to aim to outplant it by. So that is a huge success, and big thanks to all of the collaborators, like I said, specifically Moss Landing, but also our Humboldt friends who have been working on like developing this kelp culture, and no pun intended, since that's the name of our seminar today. And so then they take those kelp that they've cultured in the lab and they will seed that onto the twine, like I mentioned. And so this is Julieta holding up a little PVC pipe of this hemp twine that they've seeded it on. And so really, I'm talking about that high spatial efficiency, both in the lab and out in the ocean. And we can spool a lot of twine around these little PVC things, stick them in the tanks, and really get a lot of bang for our buck as far as space goes. So that's really neat. And then you take those PVC pipes that you've grown in the lab over a couple of months. Once they start to get a signal of kelp on them, you can see this one in this photo has like some brown fuzz on it. That's our kelp. We can bring it out into the ocean, run that PVC line over the underwater infrastructure that we've created. Again, this is a technique that we were able to learn from our friends at some of the kelp farms on the East Coast, as well as in Humboldt. Specifically, I think Kalani showed us this method. So big thanks to Kalani if you're on this call today. But anyways, this is Andrew Kim conducting this for the first time in Mendocino. And the result a couple weeks later is hopefully that we get the bull kelp to grow on the line, and that holdfast will grow over that little tiny piece of twine and then grow onto that thicker line that we've laid out in advance. And that's exactly what happened. So this photo is really exciting because it shows that we were able to get the bull kelp to grow. And as far as I know, this is one of the first documented instances of being able to culture bull kelp in the lab, then bring it out and grow it into the ocean in Mendocino specifically for bull kelp, and then get it to its reproductive state. So this was a photo that I took that was recently highlighted in a gallery. Big thanks to Josie, who's on this call today, who helped organize that and allow me to share this photo with lots of people to tell the story about bull kelp restoration. But here's a video from Bennett Bugby, who's at Moss Landing Marine Labs. And this is a video of the kelp a couple months later, and it's grown all the way to the surface. And ultimately, you can see the sore on here, meaning that it has become reproductive, which is super spectacular because that was our goal. So they're going to continue surveying this area to see if there will be more bull kelp that develops this year, because this happened last summer in 2023, and see if that bull kelp has, reproduced. Another component to all of this, like I mentioned, my research has really focused on kelp enhancement, but alongside all of this has been grazer suppression as well. And so that's been a huge part of this work, is partnering with commercial urchin divers and testing whether or not urchin suppression was necessary in order to have bull kelp restoration. And we did find in our results from this study that the grazer suppression was totally necessary and useful for the kelp to be able to thrive in its reproductive state. So with that being said, where do we go from here? What is kelp restoration looking like moving forward? We were so thrilled that that was successful last summer in Mendocino. And moving forward, although I've graduated from the lab and now have this new role at UC Davis, I just want to highlight some of the work that my friends are doing with the bull kelp this coming year. So I got this photo from Rachel Carm, and this is one of the new units that they're using out in Mendocino again. And this time they're trying to splice bull kelp into the lines, but again, elevate it up off the substrate, and also coupled with the urchin removals. So we found out last year, urchin removals, totally beneficial to be coupled with kelp enhancement, and then just trying another experimental technique for how can we best grow bull kelp and get it to stay on those lines. And so the Moss Landing has grown this bull kelp in the lab until it develops a teeny tiny nematocyst, and then they basically take it and they feed it through the line, and it's just like spliced into there, and then hopefully a holdfast will grow and it will develop and then grow just like the previous video you saw. So that's what it's looking like moving forward. So that is specifically work that's happening in Mendocino through Sonoma State University, Moss Landing Marine Labs, funded by the Nature Conservancy. But there's also lots of sea grant projects happening throughout the state and in Sonoma County that have to do with conducting similar techniques. So yeah, stay tuned for the results on that. Again, I'm not necessarily involved with that research now as I've graduated, but I do want to highlight the really awesome work that folks are doing and just give you an idea for what the bull kelp restoration looks like out here. So happy to answer questions about that if they do come up, but I do have one final section of the talk to share with you about research techniques for bull kelp restoration. So we got the storytelling. Hopefully you got that grant because you told a great story. We got the science down. It's awesome. But making sure that, throughout this collaboration and working with lots of different people, bringing different skills to the table, one of the most important research techniques that I've found for my work has been teamwork and safety. And I think it's especially important for this type of underwater science because historically kelp forest science has been kind of cavalier. And so I've always viewed it as kind of like the most extreme sort of scientific diving and work that you can do, just because there's been a lot of underwater cowboy sort of things that have happened in the world of kelp forest science. And so some really incredible science has happened in the kelp forest, but it can also be pretty limited just as far as experimental field ecology goes, because scuba diving is not accessible to people. And this field is definitely shifting because there are more opportunities for people to engage in the scope of this kind of work. So maybe you're not necessarily a diver, but other ways to get involved. But yeah, so thinking about like who we're including in these projects too is really important. So like thinking about, okay, what diversity of students are we including in our research projects and this culture of the science, but also how are we existing and working together in the communities in which we're trying to restore. So it's like the diversity and inclusivity part of the actual science itself, but then like who is benefiting from the science as well. And so I'm going to outline 4 suggested techniques that I think are important to collect better data through fostering an inclusive environment, specifically in this scientific research. But if you're on here and you're not necessarily a scientist, hopefully you can still gain some benefits from my experience as a scuba diving instructor, scientific diving instructor, and just scientific diver who's worked with many teams. So I'm going to kind of start with like the easiest low-hanging fruits for what you can do to just increase inclusivity and safety culture in your lab or your work group. And then we'll kind of work up to something that might be a little bit more complex and like harder to achieve. These are all aspects that I teach in my current role right now at UC Davis through scientific diving, scientific boating, field safety, and you can use them in lots of different contexts as well. So first, we're going to talk about equipment proficiency. And so for field science, we have a lot of different kinds of equipment, especially if you're scuba diving and maybe diving off a boat. You've got so many things to be thinking about. And so safety is really a shared responsibility among teammates. I think oftentimes it might get designated to like a PI or one person in the group to be like the head of safety, but really it's should be this shared responsibility. And we need to empower individuals to not just rely on somebody else to focus on team safety, but to focus on our own safety. And so I think equipment proficiency is really simple, but it's often overlooked because people say that they don't have the time or the money to be able to get new equipment or focus on making sure that we know how to use the equipment. And so I want to encourage you to know how to use certain communication devices like radios and spot units, things like that when you go out into the field, your emergency equipment that you might carry with you, and make sure that everyone in your group that you're working with has this knowledge just to make your team better functioning and well equipped for any emergencies that could come up. And we don't want to encourage this culture of people just being dependent on other people, really thinking about like, how do we pass down this knowledge to other people in the group? So specifically for our PIs and our researchers, I'm proposing that you dedicate the time to cover basic safety information and tools that you have for your lab. So, maybe that's on orientation, maybe that's a designated day every year or every semester that you go through this sort of equipment. For the students, I want to encourage you to ask questions about equipment. If you don't know how to use it or you're unfamiliar with it, open it up, take your things out of the box. We've got a photo of flares here that we teach for our scientific boating course, and we actually open up flares and people get to experience what that's like. Super cool. Definitely need to like make sure you notify people if you're going to test out flares so that they don't think it's an emergency. But that's the idea is, to break out that equipment and test it. We also teach people how to get into immersion suits for when they're on those cruises, so they actually know how to put one on and why they're beneficial. And then, keeping up with your first aid training as well. And then for our post-grads or our grad students or our technicians who play this interesting in-between role between, somebody who's in charge and somebody who's learning, your role is really important to facilitate information transfer between the PIs and the people in charge all the way down to the students. So making sure that everyone is empowered. Okay, so that's equipment proficiency. That's number 1. Number 2 is doing a risk assessment and briefings and debriefings for your fieldwork. And so this is really an important tool for empowering individuals to talk about their comfort levels and give them an opportunity to voice any concerns or hesitancy, or reveal gaps in whatever field plans you are proposing, and just really kind of lay everything out there. And it's a really tangible way to identify those needs and potentially like pull in all stop is what my department calls it, and just like reassess the situation. So you can also, have this, like I said, it's a tangible way to have documentation of the risks that you're incurring as a group. And so we have this system for our risk assessment that we call the GAR, which stands for green, amber, and red. It's a couple of different categories, which you can fill out a certain number based on what your feelings are for how well supported you are in each of those areas. So Jess, I don't know if you are able to share that document that I shared with you, but we should have a GAR document that you might be able to post in the chat, or we could email it around afterwards if you're interested. Just a little worksheet as something that you can do with your team to communicate what needs might you have for supervision, or what resources do you have available? What is the plan that you're about to undergo? Who is on your team and how is your team going to work together? How are you feeling individually as well as your fitness level goes, either physically or mentally? What is the environment that you're working in? How does that change day by day or hour by hour? And then what is that event and evolution complexity of whatever it is you're about to undergo? How complex is it? How many moving parts are there? And then with each of those moving parts, what are the gaps for things to go wrong? And so I really like this risk assessment because it really just puts everything out there. And it is one of the most powerful things you can give to your students or your teammates to be able to have a say in the work that you're doing. So I hope if you haven't seen this before, you might adopt it for your group. But I also have a pocket GAR that's like in my phone that I use with my friends just like for random situations. So even if it's not diving or science related, we still will just run a GAR every now and then to just like be like, all right, how are we doing with this? Like maybe we want to go on a trip. I did that with a friend recently. We ran a GAR to see like what other plans do we need in place to make sure our trip is safe. So yeah, you can have fun with it, and it's a really great tool. Okay, so that was number 2 for safety and teamwork. Number 3, a suggestion that I have is having some sort of agreement that you can have within your community, whether that's a lab space or an office space or a team space, that establishes the kind of working and learning environment that you want to exist in. And so my team here at Bodega Marine Lab, UC Davis, calls this our safe learning environment. It stands for supportive, accepting, focused, and engaging. And we give this to our students before every class that we teach, and it establishes like this valuable learning-based relationship that we want to have. So we want our relationship to be founded on making each other better. And so this pyramid that you see for our safe learning environment is like, it's a twist on Maslow's hierarchy of needs. So on the basis, it's like the most foundational things that you need. And then as you go up, other things that you can add on to really enhance your safe learning environment, with the idea being that you can't have a STEAM that's at the top without making sure that your physiological needs are met. And so this is really cool tool to like establish grounds for inclusivity, and making sure people feel valued and understood, that like you as a PI or you as a leader, care about people's physiological needs, you care about their safety and security, and you understand that they will not be able to achieve a headspace of leadership if those things are not met. So for our PIs and our head researchers, you can develop an agreement like this. And Jess, I also sent this one to you, if you wouldn't mind dropping that in the chat too, if possible, please. So you're welcome to use this and relate it to your own work and what fits your team. And then students, what kind of things do you value in a working environment? And, this could be students or this could be anybody else in a workspace. Like what kind of things do you want to prioritize and value? And then those postgrads and grad students, people that are operating in that in-between space, again, how do you foster this? How do you make sure that student needs are being met by the PI and things like that? So another thing for y'all to think about. And finally, number 4 for safety and research techniques is encouraging student leadership and leadership among individuals. And so really making sure that we're passing knowledge and skills along to continue this type of research. So For bulk help research specifically, like there's so many different things that are changing from year to year, and making sure that people are having the ability to carry on that research and it's not all just resting within one individual like a PI or a lab tech or someone, but passing on the knowledge. So for PIs and researchers, this looks like encouraging students to run the risk assessments, to do briefings and debriefings with your team, to be in charge of communicating things is a really important empowering tool. For students, you can ask for opportunities to learn under supervision. So if you want to better learn how to drive your boat, or you want to learn how to service some sort of equipment, making sure you're asking for those is really important. And again, for our post grads and our grad students and our techs, mentoring other students in low stake environments. So when the opportunity comes to be able to mentor someone in a technique or a scenario without it being super high pressure, that's a great opportunity. Also making those opportunities for each other to establish that proficiency. So with that being said, we just talked about three main research techniques. We've got science, we've got storytelling, and safety that I think have all led to some really successful bulk kelp restoration work in Northern California that I'm so grateful to be a part of. And these things are all really important in increasing inclusivity and inclusion in this story of kelp restoration that we all care so much about. So we want to celebrate the diversity. We want to celebrate the interdisciplinary approaches to this. And we also want to collect really good data and have good stories. And so with that, some acknowledgements from a lot of the wonderful people I've worked with. And this is totally not an all-inclusive list, but just, yeah, I'm really grateful for this community. And thanks for having me on today. I know we're like right at the time limit. So I'm happy to like stay on and answer questions or answer questions via email.